Well, thanks, Leila and Carrie for setting this all up. And I'm going to get into a little bit of the research that we're currently working on. And so, our primary goal from our current work is to produce genetically diverse Dutch elm disease-tolerant American elms for use across the urban and rural forested landscapes. So some of the subcomponents associated with this include identifying additional Dutch elm disease-tolerant American elms for the horticultural community. So these are genetically identical clones of trees with good characteristics like this, good growth form. We're also interested in identifying additional DED-tolerant American elm for seed orchards and restoration plantings. So elms for seed orchards need to reliably produce a large number of progeny that have good disease resistance. And the seeds from these seed orchards could be planted in nurseries or broadcast in the natural systems to produce material for restoration or used directly in restoration. We're also interested in testing resistance of Dutch elm disease-tolerant American elms to different strains of Dutch elm disease. So we've collected fungal pathogens from around the United States, and we've assessed them we're interested in assessing them for differential pathogenicity. We're also developing guidelines for reintroducing elm to rural and urban areas. We're working with collaborators to identify genes responsible for DED tolerance. One of the big things we're working on, too, is streamlining the testing process, so coming up with early screeners and many more research here. So we've been working with a variety of partners to identify large survivor American elm trees, which may exhibit Dutch elm disease tolerance. So, for instance, we have great partnerships with Christian Marks at the Nature Conservancy, and he's been critical in sourcing elm material from the northeastern U.S. We've also been working with Dale Lesser, a farmer and elm enthusiast from Michigan, who has sourced material from the lower Midwest. And our partner here at the USDA Forest Service, Kathleen Knight, has worked with our tech gurus to set up a website in which users can submit information about large survivor elms. So since its inception, this database has amassed submissions spanning the historic range of elm, as you can see, right? I think we're closing in on nearly 900 submissions now. So when we pulled this map earlier, so we've got a decent repository of material to start if we have the time to do so. And so once we identify material of interest, we collect it during the spring. And so the image here on the left shows an arborist in New England collecting elm scion or new growth, which is immediately boxed up and overnighted to our lab for vegetative reproduction and pollen harvesting. So most recently, we've been working with a residual bank of elm material and survivor elms from New England, Michigan, and Ohio, which were propagated from 2016 to 2017. So once received in our greenhouse, we use various propagation approaches such as bud grafting, shown here on the left, to produce clones or identical genetic material, which is grown up across plantations and allowed to mature. We also utilize pollen exclusion bags to conduct controlled crosses by hand pollinating Dutch elm disease tolerant material with newly harvested pollen collected from these survivor trees. So following seed set, the seed is collected and either planted in the greenhouse and transferred to a research plantation or stored for future use. After trees grow for 5 to 7 years, we inoculate them with a 50-50 mix of Ophiostoma olmi and Nova olmi and visually assess symptoms for Dutch elm disease-induced canopy decline. So these largely include foliar yellowing, browning, flagging, and leaf drop. So the results from a lower Midwest survivor elm study here in the Delaware research plantations is shown here on the right. And so you can see the unique elm genotypes or the selections along the y-axis and the mean 2-year post-inoculation Dutch elm disease-induced canopy decline on the x-axis. So note we've shaded the 2 Dutch elm disease tolerant controls here in black, and so these are Valley Forge and Princeton, as well as our known Dutch elm disease susceptible selection in red, which is American 57845 here at the top. We also have new selections from the lower Midwest that are highlighted in green and additional crosses in gray. So what you should take home from this figure is that some of the new selections from the lower Midwest perform as well as commercially available Dutch elm disease tolerant selections. We were also approached by a silviculturalist, Gary Swanson, on the Chippewa National Forest in northern Minnesota to see if we could assist them in creating Dutch elm disease tolerant American elm for use in restoration settings. And so, as these systems are kind of exposed to some harsh climatic conditions in the northern U.S., these individuals needed to be cold tolerant. So for this, we collected pollen from 4 survivor elm trees on the Chippewa National Forest and crossed them with Dutch elm disease tolerant American elms, and we planted this material across 4 plantations, 3 in Minnesota and 1 in Ohio. So our results from this study suggest greater survival and growth of material in Ohio relative to Minnesota, as we expected, and this was largely driven by harsh environmental conditions in Minnesota. So you can see here in this figure where we have differential Dutch elm disease-induced canopy decline between these regions, with material planted in Ohio denoted with gray bars and material planted in Minnesota denoted by black bars. And so the results of this kind of indicate a gene by environment interaction, which highlights the potential need for locally adapted stock for unique restoration activities. And one of the other primary areas of focus, as Leila mentioned before, is the reintroduction of American elm to the rural to urban gradient. So here we've selected this quote from David Richardson to frame our use of elm in restoration. So if ecosystems are viewed as open and dynamic, recreation of some historic condition becomes an exercise in nostalgia. So folks familiar with the elm, or with elm prior to Dutch elm disease outbreak may recall this species as a foundational species across both riparian forests and heavily utilized in urban areas of the eastern United States. So furthermore, many have fond memories of the graceful canopies of American elm trees flanking urban streets. So despite the previous abundance across the natural landscape and its heavy use in urban systems, the use of American elm moving forward should be in conjunction with non-elm species to sustain or enhance ecosystem function and resilience. Furthermore, urban foresters well know that monocultures can be rapidly wiped out as pests or pathogens move in, resulting in unintended, undesirable conditions. So here we have 3 primary foci with our elm restoration work. And so we're working in emerald ash borer impacted floodplains, formerly grazed riparian corridors, and reclaimed mine lands. So recently, folks have been using American elm in conjunction with some EAB impacted systems up in northern Minnesota, and so they're looking for replacement species for these black ash wetlands in northern Minnesota. So emerald ash borer is kind of on the precipice of reaching that region, and so they've been conducting some manipulative experiments there with thinning, girdling, and restoration approaches. And so here they planted individuals of dozens of species and found that American elm was one of the best performers in these systems, despite the exposure to seasonal water inundation and harsh winter temperatures. So additionally, an article here by Lewis Iverson and colleagues emphasized that although several other species exist that could be used in restoration of EAB-impacted black ash systems, the climate envelope for many of these systems, such as quaking aspen, is shrinking, and they may not be suitable for future conditions. So American elm, on the other hand, may be very well suited to colonize areas vacated by black ash. So unlike natural forests, trees in urban forests are exposed to harsh environmental conditions such as soil compaction, low soil quality, or contaminated soils, urban heat islands, construction damage, vandalism, competition from invasive species, and many more factors. Furthermore, these trees must meet aesthetic and structural requirements. They must exhibit high survival and high levels of disease tolerance. So now I'll kind of fill you in some of the more urban experiments that we're working on. So we have a new experiment that we've just deployed in tandem with other species, where we've utilized elm as our backbone species in some urban riparian forests across Columbus, Ohio. So as you can see here from the image on the left, these systems are inundated with invasive species, in particular bush honeysuckle. So this experiment has been deployed across 5 locations in Columbus, Ohio. And in collaboration with partners at Ohio State and the City of Columbus, our overarching goals for this work are to control invasive shrubs while simultaneously enhancing biodiversity and habitat quality. So by removing invasive shrubs, we seek to enhance ecosystem function and integrity. Additionally, we're investigating the relationship between vegetative structural complexity and avian abundance and behavior, We're also interested in reintroducing native plant material to these understories and determining strategies and guidelines for riparian restoration. So below here you see a schematic of the experimental layout with 2 plots assigned to an invasive removal treatment and 2 plots designated as non-removal controls. So 1 plot from each of these treatments was further assigned to receive an underplanting of native woody material. And for folks who participated in or undertaken manipulative experiments in urban areas, you're likely well aware of the challenges associated with these activities. For instance, the use of chainsaws in high traffic areas, as you can see here by the photo on the left, and the need to temporarily close high traffic multi-purpose paths can be negatively perceived by the public and create some roadblocks that we have to navigate along the way. Additionally, here we have a series of images associated with the deployment of this urban riparian restoration work. Specifically, on the left, we have an understory cleared of invasives, and then in the middle here, we have the recently planted restoration material, and on the right, a close-up of some of the deer cages we've deployed to deter herbivory. So we're in the process of evaluating survival and growth of these planted seedlings. We're assessing vegetative dynamics across time and space and assessing changes in canopy structural complexity and their implications for urban wildlife. And as you can see from this image here, the urban wildlife, particularly deer, are prolific. So finally, in collaboration with a myriad of Forest Service scientists, university partners, and municipalities, we're deploying a new experiment to test newly developed Dutch elm disease-tolerant American elms across the urban and urbanized landscape. So despite the robust performance of some of these selections in our research plantations, most varieties have not been tested for tolerance to urban stressors. We've identified and received approval to plant elms across an urbanization gradient across 3 cities, Columbus, Ohio, Philadelphia, Pennsylvania, and Newark, Delaware. So the primary goal of this study is to investigate how urbanization metrics and a variety of other factors drive elm tree establishment and growth. Some of our future directions are focused on continuing to identify and test potentially Dutch elm disease-tolerant material to enhance the genetic diversity of DED-tolerant American elm. We're also investigating differential virulence or pathogenicity of different strains of DED from around the U.S. We're interested in studying gene flow as associated with the Dutch elm disease-tolerant restoration plantings and the gene flow into these natural systems. We're also continuing to work with partners to investigate Dutch elm disease-tolerant resistance genes. And finally, we're working to refine early screening tools to increase DED tolerance testing. And with that, we'll end with some acknowledgement of our partners. Specifically, we've been working very closely with the Manton Foundation, who's funded most of this recent work, Dale Lesser, an elm enthusiast from Michigan, Christian Marks and the Nature Conservancy, Columbus Metro Parks, Friends of the Lower Olentangy Watershed, the Finger Lakes National Forest, and the Chippewa National Forest, as well as the U.S. Forest Service, state and private forestry, specifically Linda Haugen. And with that, I think we'll take some questions.